It's a pleasure to welcome Dr. Christopher. This is very exciting for us as African manufacturers because we are going to talk about two things. We are going to talk about platform technology and then how this platform was used to develop malaria vaccine. So my job here is just to introduce the speaker who is for me to discuss how this technology, certain technology can be transferred to African manufacturer. So Dr. Locher is the CEO and co-founder of Versatope Therapeutics, a clinical stage Massachusetts-based company that is currently focused on the development of immunotherapeutics using nanovesicles. He previously led drug discovery projects at Vertex Pharmaceuticals in Boston, inflammatory diseases at Opsona Therapeutics in Ireland, and malaria and alpha viruses vaccines at Maxigen in California. So over to you, Dr. Locher.
Thank you, Simon, and pleasure to be here. Yeah, I'm happy to share this information that has been a National Institutes of Health sponsored program. It was a grant to first focus on a transmission blocking vaccine candidate, PFS 230, that blocks the transmission of the oocytes in the gut of the mosquito and prevents the life cycle of malaria. We then applied the technology to CSP for a bispecific vaccine, and we have a candidate that expresses both CSP and PFS 220. Today, I'll tell you a couple of different forms of those and how we use these potent little nanovesicles that are about 100 nanometers in diameter to develop new vaccines and immunotherapeutics. I am the principal investigator on the NIH-sponsored grant. Kevin Wyant is the principal scientist, and Jessica Lee is my chief development officer, and a retired colonel, Anzima Men, has been our chief medical officer, and we've been supported by several scientists to do this work. We are a small company. We're only 11 employees based in Lowell, Massachusetts, just outside of Boston. We were founded about 8 years ago. Since then, we've been focused on a influenza vaccine, a universal influenza vaccine, and we licensed the technology from Cornell University. It's a technology that expresses recombinant polypeptides on the surface of bacterial vesicles. And also we have a single dose formulation that I'll tell you a little bit more about. The malaria vaccine candidate, like I said, was supported by an NIH grant. We do have CSP expressed on the surface of the vesicles. We do have a single dose formulation. It can be lyophilized for a room temperature stable form, and that's currently under development in accelerated stability studies. At 37 degrees, it's stable for at least several weeks and months at room temperature at 25 degrees. This should be available for clinical development relatively soon. For our phase 1 clinical study, we've designed it and discussed it with the National Institutes of Health. It's not going to be a placebo-controlled study. We will do 2 or 3 doses with 2 or 3 immunizations and evaluate safety and immunogenicity as the primary endpoints. So as you know, vaccines are a hassle. They do not last a long time necessarily. It requires significant boosting, and the immunity to diverse strains, or in this case with malaria, to diverse targets in the malaria life cycle, are not always the most effective. So we want to develop a vaccine for malaria that can focus on not only pediatrics, but also young adults as well, or adults, and something that is more immunogenic. People don't like injectables generally, and they can be expensive. The manufacturing process can be costly, and they do require often a cold chain dependency, certainly with the case of messenger RNA vaccines. So there could also be complications due to adverse reactions with lipid formulations and allergies in some people. So we want to get around that. With our universal influenza vaccine, we've developed a full clinical program. It is a large market opportunity, and it captures genetic diversity on a single nanovesicle. We do this through genetic engineering by expressing different epitopes in tandem, one after another. And in doing so, we've been able to show that we can get protection and reduce viral loads with an off-the-shelf solution for pretty much all variants of influenza A by focusing on a very conserved target called matrix 2 protein that's clinically validated and market tested. So we have developed in this process optimized cell lines that are available. We've engineered these microbial cells. They are E. coli in origin. They are derived as probiotics. They are biosafety level 1, so they are not a pathogen affecting humans. And that's a key differentiator between what other people are doing and what we're doing at Versatope. In addition, our products are a millionfold less pyrogenic than the wild type. This is because we've genetically engineered them so that the lipopolysaccharide in the membrane is no longer biologically active and actually serves as an antagonist to toll-like receptor 4. This is another very key distinguishing feature between Versatope's technology and other outer membrane vesicle technologies that are available. It is versatile. We can express polypeptides on the surface, both epitopes for vaccines as well as paratopes for antibodies for cell targeting. That's why we called the company Versatope. We also have a conjugation step, a simple chemical conjugation step that other people have used for malaria vaccines, such as PFS 230 as well. By using our recombinant expression system, we have very high density display, up to 1,000 different antigens per vesicle. Like I said, they're thermostable at room temperature. We have a single dose formulation by using polylactide coglycolic acid, or PLGA for short. It is a sustained release microparticle that avoids the repeat immunizations. We do have an FDA approved process for both our GMP manufacturing as well as our 24 release assays for analytics that I'll talk a little bit more about. There are clinically approved outer membrane vesicle vaccines. One is known as Bexsero. It's a meningococcal B vaccine that's developed by GSK. It was developed in Italy by the group in Siena under the direction of Rino Rapoli and Alan Saul, who got this product licensed in the United States several years ago. So it is market tested and proven that one can develop a product using outer membrane vesicles. But there are different features about this, primarily because they use an alum adjuvant, which serves as a slow depot or release of the proteins that are co-administered with the outer membrane vesicles. And this is because their outer membrane vesicles are relatively high in pyrogenicity. They will induce reactogenicity and a bad reaction. So they're required to use alum adjuvant to slowly release the product as a depot adjuvant over time, so you don't get a bolus injection or a bad reaction at the site of injection. So that's a key feature of what we don't do. We don't use alum adjuvant with our product. In fact, there's a number of different advantages over other adjuvants. We have multiple toll-like receptor pathogen-associated molecular patterns, so TLR2, 4, 5, 7, 8, and 9. So it has a very potent immunostimulatory properties and a diverse profile. They're thermostable. They get taken up and target the lymphatics. They have reduced reactogenicity compared to other products, and they are suitable for mucosal or dermal delivery as well. There are other advantages about why one would use these. This is because these physical chemical properties can be fine-tuned with genetic engineering. They've been evolved over years to penetrate through tight junctions of cells and get taken up by the lymphatics to modulate the immune system. They are more resistant to osmotic gradients, which is different than liposomes, and their sizes are quite a bit smaller than liposomes as well, which enable them to have high penetration of tissues. In addition, multiple routes are feasible. This has been shown by not only injectables, but sublingual and oral tableting of the vesicles is also feasible. Intranasal immunizations for clinical studies are ongoing, as well as transdermal patches have been proven in preclinical studies. Their increased stability in serum permits lower doses. They are relatively inexpensive in terms of raw materials for manufacturing. We have an established process that is approved and scalable, and they can be easily lyophilized and just resuspended in water because they are made primarily out of polysaccharide. and the polysaccharide and the sugars in the vesicles enable them to be very stable. I do have a robust team. So myself, I founded the company, and I have a chief medical officer, Anza Mamen, who has done thousands of subjects in clinical studies, in really dozens of them, while he was working with RARE with the U.S. Department of the Army. Jessica Lee, who used to work at Merck for many years and has guided many phase 1 through phase 3 global clinical studies, primarily in vaccines. The Nobel Prize laureate, Drew Weissman, is based at the University of Pennsylvania, is on our scientific advisory board, as well as our key scientists that have led this work. There is a publication that is available online that talks about the different methods of macromolecular display that was published by the Cornell Group, primarily Kevin Wyant on our team. And our preference is to display these with recombinant technology. And this is because it's relatively easy to induce and express the polypeptides, and you make one single product. There are no chemical conjugation steps using biotin or docking with antibodies, but these other approaches are feasible, and this is something that we originally tried with CSP, the circumsporozoite protein of malaria, as well as PFS 230, and our preference is to do the recombinant display, and I'll tell you why shortly. So we are clinical stage. We are raising money for our clinical study for our universal influenza vaccine. All this work has been supported by the NIH, National Institutes of Health, but given the new changes in the federal government in the United States, we've been faced with funding challenges over a year now. So briefly on the bispecific malaria vaccine, we can chemically conjugate these. We've done this with chemical conjugations. We find that they actually get very potent transmission blocking anti-malarial antibodies using this approach. And there's also a lower complexity and lower cost using Advidvax. And we find that the chemical conjugation worked a little bit better for transmission blocking, or in the case of CSP. Briefly, we know that we can get very high endpoint antibody titers with our malaria antigen with PFS 230. We've also used this approach for neuraminidase, and we know that the neuraminidase will block the enzyme activity when we conjugate these to the malaria vesicles. So we do get very high endpoint titers of over 1 million in very small rodent animal models. Our new strategy that we've tried using recombinant display, which is our preference, is we can express domains of PFS 230 or have a repeat circumsporozoite protein peptide for targeted immune responses. And this is because we can scale these up and purify them in one go without having to do the chemical conjugation steps with PFS 230 and CSP. We've used this approach for our malaria vaccine, for our influenza vaccine, that is a clinical candidate. We have 6 different antigenic variants or different epitopes that are displayed in tandem. Each epitope is about 30 amino acids and separated by a flexible linker to enable accessibility on the surface of the bacterial vesicle. It's anchored into the membrane using a transmembrane protein called CLIA. And this anchors the polypeptide and displays it on the surface of the nano-sized vesicle. As a result, we can get immunity to multiple different strains of influenza. There are 6 different phylogenetic groups in influenza that are associated with the matrix 2 protein. So we've captured the genetic diversity of influenza all on the single nanovesicle. And this is anchored into the membrane using CLIA, which also serves as a minor adjuvant in its own right. So we've tried this approach. We know that we can get very high levels of proteins displayed on the surface. We can get thousands of copies of proteins, and having this high density of display enables us to get very high endpoint antibody titers. We do get diversity to diverse strains in this approach. And it's overall, when you go back to thinking about basic immunology, it is these are hapten-like epitopes that are conjugated to a nanovesicle carrier. So in the case of influenza and in the case of CSP, these are primarily B cell epitopes that get stimulated and you get very high endpoint antibody titers. They are induced, they do induce innate immune responses quite potently. You can measure cytokines and innate immune responses and the different functions. After phagocytosis, it increases chemotactic cytokines and chemokines, as well as cytokines and macrophages that are pro-inflammatory, primarily a Th17 response, and this can be measured as well. And there's a really nice review that was written several years ago about the host immune response to these little nanovesicles. As I said, Cly-A is also an adjuvant in its own right. It will stimulate uptake by the vesicles. So we find that when we use our Cly-A, we get very high endpoint antibody titers. So it is a good fusion protein that one can use for display on the surface of the vesicles for not only anchoring the polypeptides on the surface, but also stimulating the immune system. By transmission electron microscopy, you can visualize Cly-A as a pore. It is an ion channel on the surface of the bacterial cell, and this is the outer membrane vesicle that gets secreted to the surface and into the supernatant of the bacterial supernatant of the culture when we do a bioreactor reaction. And you don't see these pores when you use empty OMVs. It's just a basic transmembrane vesicle that one can see here, which we call empty OMVs. In terms of our candidates, we see that the CSP actually has better immune responses and certainly on par with neutralizing monoclonal antibodies at our ability to reduce the liver burden in mice for circumsporozoite protein. And it also improves survival with circumsporozoite protein with our lead candidate shown here in green. And this is again comparable to a monoclonal antibody. In terms of PFS 230, the chemical conjugate also induces strong transmission blocking antibody responses. In fact, it outperforms the PFS 230 if it's just conjugated to OMVs or when it's combined with alum adjuvant. So this is why the NIH supported this program, because it is an alternative for an alum adjuvant type of approach when we develop malaria vaccines and other vaccines in general. In terms of a scalable manufacturing process, we have developed a full process that has scalability to 40 liters at using cGMP. We get about 5,000 doses if one uses 20 micrograms per dose and about 2,000 vials from 40 liters. So it's suitable for a phase 1 and a phase 2 clinical study with just using a benchtop bioreactor. It is a process similar to brewing beer. We do have 24 qualified assays and 11 for information only that the Food and Drug Administration in the United States has approved for clinical use and for release. So we have allowance to go to the clinic using our process. We also have a spray-dried system using lyophilization. And like I said, we've shown stability at room temperature for our products, and which leads to better economic development, so you don't have to continue to make new batches of vaccines and store them in the freezer. We do have excellent reproducibility in batch-to-batch manufacturing, and our testing has shown that extended batch manufacturing is feasible. We do have a very favorable cost of goods because we have high yields, and we can use a basic broth medium in simple microbial bioreactors, or even at small scale, you can do these in shake flasks as well. They are portable in terms of a scalable equipment. One can put this on a skid, so it is available for decentralized manufacturing in small microbial bioreactors. So briefly, to get a bird's eye view of the process, we use shake flasks for an adaptation phase when we have working cell banks stored in the freezer. We're able to take the cell banks out. We adapt them to a select medium through a couple of processes over 48 hours. And then we inoculate our bioreactors on the benchtop, and we're able to scale these up and then harvest them. Our manufacturer does 40 liters, but we have 15 or 30 liters that we use in-house. Our downstream process is relatively simple. We then remove the cells either by harvesting. One can use centrifugation or filtration, hollow fiber filtration. The TFF step is part of the downstream process where we can concentrate and filter it. We do have a nuclease treatment step that we remove the host cell DNA using nucleases. And then we remove the nucleases by chromatography and are able to further purify the outer membrane vesicles. And then finally, for sterile filtration and fill finish for either lyophilization or put them into vials for storage. We also have in-process monitoring of our upstream bioreactor process. We find that when we induce the polypeptides with a sugar that activates, it's a plasmid expression construct, and the plasmid induces the protein gets expressed on the surface of the vesicle, and over time, it can produce, and there's an optimum window for harvesting the vesicles before they reach the plateau here when they're still in the log phase of growth, and then they'll hit like a lag phase. One can continue to feed these and harvest them, but the optimization by measuring the target ratio of target antigen to the vesicle that's still intact is important because you don't want to get lysis of the vesicles, which happens toward the end of the culture period. So one wants optimal optical density in the bacterial culture, optimal protein, and optimal outer membrane vesicle formation. And we can monitor this with SDS-PAGE. We have reporter antigens for both outer membrane vesicles as well as our target proteins. We use size exclusion chromatography on an analytical scale to measure the vesicles in the process as well. And then we have other monitoring such as SDS-PAGE, reverse phase HPLC, and again, NTA and transmission electron microscopy, which is a nanotracker analysis to actually count the number of particles. And one can do this in process as well. So it's important to not let the cultures go too far. If you let them go too far, there's a lot of lytic material and you have a poor quality product. But if you harvest them early, while the culture is still relatively healthy and you have the target protein affiliated with the vesicle, then you have high quantities of particles and high quality of the antigen as well. We have a whole battery of critical quality attributes that we've used in our analytics. Like I said, analytical size exclusion chromatography. We use SDS-PAGE for our target proteins. We're able to measure the proteins by densitometry. We have reporter proteins as well as our target antigens using immunoblot. We've done mass spec profiling of the vesicles. The major protein is in fact our protein of interest using mass spec. It's about over 50% of our target antigen overall. And we've also done ELISA, reverse phase UPLC, mass spec. We have a monocyte activation test. We also do the Limulus amoebocyte lysate test for endotoxin. And we can measure the number of LPS molecules using gas chromatography followed by MS. To measure the host cell DNA, we use qPCR. We use gel electrophoresis and picogreen as an ELISA. These are commercially available, the picogreen tests. And then to understand the size of the particles in the vesicles, we can use nanotracker, dynamic light scattering, TEM, as well as cryo-EM. So, like I said, our vesicles are about 1,000,000-fold more attenuated than the wild-type nanovesicles. This was shown in our monocyte activation test, then using human PBMCs, or peripheral mononuclear cells. And this has a interleukin-6 response that measures endotoxin. And when you do this with the human cells, it serves as a read and is acceptable by the FDA and the EMA as a endotoxin release test. So we're the first company to use the monocyte activation test in the United States for release without having to rely on the LAL, which the FDA is seeking alternatives to. So it's 1,000,000-fold less, and it actually serves as an antagonist to toll-like receptor 4. This is because there's only 4 carbon chains or acyl chains, these long chains that are affiliated with the lipopolysaccharide. And because there's only 4 chains, it doesn't engage or dimerize the toll-like receptor 4 and have that engage with CD14. So it actually blocks the signaling of toll-like receptor 4, and you get very low levels of the IL-6 production. In terms of scalability, we do know it's economical. It is feasible to do extended batch manufacturing as well. At our current scale, these are all based on ballpark quotes from contract manufacturing organizations. We do prefer the lyophilized form so that you can keep the product and don't have to make new batches. But it costs a few $1 million just to do the first batch and to develop all the assays. But as you scale it up, it gets to be less than $1 a dose. Certainly, probably fractions of $1 for commercial scale if one uses extended batch manufacturing and lyophilization. So in that case, it's really ideal for vaccines that require very large volumes of product. And you don't need huge bioreactors to make this. This is because you can get a lot of doses in a relatively small scale. But in addition, even with a 2,000-liter bioreactor, one can do extended batch manufacturing and get probably a lot of different doses, millions of doses with this process over the course of 1 week in 2,000 liters. So it is, like I said, available for decentralized production. One can make a portable cGMP bioreactor. There are wave systems that are relatively small that are portable. We also have you can put all this on wheels and have it on a skid. That includes the not only upstream process, but the downstream process as well that can be shipped abroad and can be used on site for the development of vaccines at the site of an outbreak. So this is important for pandemics or epidemics that emerge in specific regions, and you have a solution here that's available at a relatively small scale to address an immediate need. I'll just shift gears a little bit and talk about our single dose formulation using PLGAs. This is also done at a relatively small scale. One can concentrate your product in PBS. You can have the chemical solution of PLGA and dichloromethane and form an emulsion, and then add polyvinyl alcohol to it to form microparticles and evaporate the dichloromethane overnight, and then centrifuge and lyophilize the product. This is an established process. Clinical products have been made for this, and these do make the vaccine available for multiple routes of administration. This is for our single dose microparticle formulation to allow a slow release of the product. It releases it over about a month, more or less. And work has been published by the Cornell team initially. So you can actually measure the total protein released over several weeks at 37 degrees in just a PBS solution. And you can also measure the total protein by antigen, and that's slowly released as well over time. So we've done this with immunizations for circumsporozoite protein with malaria, and this work is ongoing here at our company. The particles actually are larger than the nanovesicles themselves. The nanovesicles are actually contained within the microparticles, and most of our work has been done using intramuscular injection, and there's a publication available several years ago that was published in Vaccine showing that the single-dose vaccine actually worked as well as a prime boost approach and providing 100% survival in a mouse animal model. This is extended over 6 months of time. So this can, in theory, provide close to a lifetime of immunity using a single-dose formulation. So we're in the process now of doing additional work with this PLGA formulation for single dose. Not only for our products for influenza, but also for malaria. And we're doing this by comparing different adjuvants. The VT105, we know it's stable after lyophilization with our product at room temperature over several months. We're out to a year now, and it is stable. So it's stable indefinitely at room temperature for all intents and purposes. It does retain its stability. You can visualize this on a gel. There's really no change and there's no degradation in product after it's been lyophilized. And you can visualize the vesicles after they've been reconstituted with distilled water, and there's really no change in the vesicle properties either. So this product and these vaccines do support our key objectives for the World Health Organization. We do know that we want to meet our targeted profile of it having an effective vaccine that reduces blood stage infection over 12 months. We do know that our product outperforms the approved RTSS vaccine in a luciferase model and in the passive transfer model. It is comparable to other vaccines. We've seen no safety reactions with our influenza candidate in GLP safety studies in rabbits. This has all been approved by the FDA, and there were really no remarkable findings in these products at all. It meets the target requirement for being affordable. We do know we can make it for less than $1 a dose at commercial scale. And our current processes can increase yields quite high, more than tenfold than what we've originally approached with our influenza vaccine. We do want a single dose formulation as well. That's our preferred PLGA microparticle formulation, but we do know that 2 or 3 doses could be acceptable for lifelong immunity, but not really ideal. We do get very high endpoint antibody titers, and although some of this work was outside the scope of the SBIR, we have continued to do some of this work. So in terms of a target product profile, we do have a minimum and preferred characteristic for the vaccine product in terms of the route of administration, indication for emergency use authorization, target population in pediatrics, but we prefer all age groups. We want our safety and reactogenicity, efficacy, schedule, onset of immunity to be relatively robust. And our preference is either a single dose vial or a single dose administration with the vaccine. It's possible to do a tablet for sublingual immunizations, but more work needs to be done with that. So in terms of clinical study costs, one could do a phase 1 clinical study for about 60 subjects for about $2 million. We have a minimum and a maximum that we've projected for this, and we've shared this work with the NIH. They asked us to pull together a clinical plan of what this would look like and what our field studies would look like. So it does get very, very costly when one gets to a phase 3 clinical studies. So this is all work that would need to be done in collaborations with non-government organizations as well as government organizations that want to see a malaria vaccine implemented in the field. So there are additional publications available on our website. If you're interested, feel free to read these. There's plenty of material out there. So in summary, we do have unique strains that are available for hypervesiculization, or they make a lot of product, more than 500-fold compared to the wild type. This has been shown. Our products are very safe. They're 1 million-fold less pyrogenic than the wild type vesicles. They are versatile. We can express different payloads and combinations of payloads. We do have high density display, and we do have a thermostable formulation following lyophilization. So what makes us different is that we believe we can have a broad clinical impact for not only infectious diseases, but other diseases as well. We believe that economically we have a superior value proposition. We have strategic differentiation with low toxicity, no other adjuvant required, room temperature stability, and a vaccine that is relatively potent, targeting diverse aspects of the malaria parasite life cycle. We have an adoption differentiation as well by alleviating different stages of the malaria parasite, as well as influenza and other diseases that could be associated with other comorbidities, and thereby reducing overall the disease overall. And not only the disease itself, but other secondary diseases that are associated with it. It does support global health priorities, and we do believe we have a long-term sustainable solution. So I'll stop there, and I'm happy to entertain any questions you may have.
Well, thank you very much for this very exciting presentation. I think I'll open this for Q&A. So if you have questions, you can raise your hands or put it in the chat. So let me start. I mean, from your presentation, 40 liters of fermenters gave you about 5,000 doses of the vaccine at 20 microgram. So, and then you said 2,000 liters we give potentially millions of doses. So how is that possible? Again, you compare the vaccine with the GSK barracks. You know that they think by 2028 they will be able to bring down the cost to less than $5. And this is going to be just about $1 per dose, and we're looking at single dose. So the translation is not clear to me from 40 liters or 5,000 doses to 2,000 liters. And then finally, you know, the key thing is the partnership with African manufacturers. So what sort of relationship or technology transfer do you, or are you envisaging with the African manufacturers vis-a-vis the products? You know, all the preclinical studies have been done, and then clinical development is what is remaining to be able to commercialize the product. So is it going to be like co-development with the African manufacturers so that together the clinical trials are done from phases 1 to 3? And then, or is Alpha licensed to African manufacturer, and African manufacturer has to deal with how they can conduct their clinical, proceed with their clinical development. Over.
So I'll take the first question first. So going from just 2,000 vials at 40 liter scale to millions of vials going at 2,000 liters is primarily done with extended batch manufacturing. So one can, I've talked to manufacturers about this, and that's where you feed and you harvest over a period of time. So you can take off a fair amount of the supernatant and remove the cells and keep feeding the culture over a single culture without taking down the bioreactor process. This is something we haven't done at the 40 liter scale. This is just one batch over the course of a few days. We do a single batch, we harvest it, and we're able to get 2,000 vials at 40 liters. We reckon that with extended batch manufacturing, we can get much more from that and extend the batch for, I don't know, a very long time. I don't think a month, but probably at least over a week, at least 10 days by doing consecutive harvests. And in doing so, if one can put down lyophilized material, you can get even more material over time because you're not continually making fresh batches. So it is feasible to do this at 1,000 or 2,000 liters after discussions with manufacturers. And this is feedback I've had from people that are doing this at commercial scale. This was based on their recommendation. In terms of clinical studies, yeah, this is something we can discuss. We don't have specific commitments right now for doing clinical studies. This is just basically a study that we've talked about with the NIH by doing a simple phase 1 clinical study. We could do it here in the United States for the phase 1, but we would need to do other studies abroad for the phase 2 and beyond. Certainly doing field studies is not something that would be realistic to do in the United States. There are several vaccine units that could be done in collaboration, but we would need funding for this approach. And this is something that could be done as a collaboration or a licensing agreement, and all that needs to be worked out in the future. We do know we can tech transfer our process. That's not a bother. When we made our first batch at 40-liter scale with our manufacturer, they did it in one go. We transferred the technology from our 30-liter bioreactor. They gave it a go. We made an engineering batch. There was no problem. They followed our protocol. They got it made. There's no problem. They made the GMP batch. That was no problem. So just in a couple of batches, we were able to get a GMP process. So I'm not concerned too much about the technical capabilities. It does require some adaptation of bioreactors and process. So it just depends on the manufacturer and their equipment and what they're able to do with what material they have in-house. So it is a microbial bioreactor fermentation process. So if one has that equipment in place, it can be easily tech transferred. You're on mute, Simon.
The way we work is that, the way we work, is that, of course, the manufacturers that are listening to you, if any is interested in the technology, they will contact the secretariat, AVMI secretariat, and then we'll put them in touch with you, and then you can discuss the details of how you want to proceed, whether it is tech transfer, outright tech transfer, or co-development, or whatever sort of arrangement that you may do. So that's how we can proceed as far as AVMI is concerned. So any other question from, yes, ED. Oh, okay.
No, I have no comment. I have another meeting, so I was just saying that I'll be leaving soon. Thank you very much, Chris. This has been a very, very interesting discussion, and I hope some of our members will reach out to you to take this forward. Thank you. Over to you, Simon.
You're welcome. Thank you. Pleasure.
Thank you. So any other question or comments from the participants? Great. So thank you again, Christopher. I look forward to visiting your facility during the summer. I hope to be there by next week. So I'll come visit you and then see how we can take this forward.
Very good. You're very welcome, Simon. It'd be a pleasure to have you.
Great. Thank you so much. And thanks all the participants. And if you want to proceed with this collaboration, please do let the Secretariat know. And then we will definitely put you in touch with Dr. Locher. I think Kumar, you can ask your question. Go ahead and ask your question. Hello, Kumar.
He's typing it.
Yeah, I guess, okay. What is the stage of development is completely done?
Yes,
Yeah.
the influenza vaccine is clinical stage, and the malaria vaccine is still preclinical stage. So we do have the safety tox work that remains to be done, and the optimization of the single dose formulation. So this is something that we're currently working on, and it could be ready in the clinic as early as next year, if all the funding and all the resources are available. Yes.
Okay, great. Thank you so much. And then
Very good, Simon.
You'll be hearing from our members soon. Thank you. Appreciate it. Bye.
You're welcome. Thanks. Bye-bye.
Thank you.